Hello students, as promised I have put together an introduction to vaccines and I will just briefly define the traditional as well as some of the modern techniques that are being developed. And the nice thing about this is actually most of the slides, first set of slides, I pulled out of the lecture that I give later on host resistance to viral infections. So let's see, I think it's chapter 5. So we'll cover it now, and then I can be more focused during that lecture when we talk about the immune response. And when we do talk about the immune response, you'll see the mechanistic basis for vaccines. So just an overview, the purpose of vaccines is to stimulate the production of antibodies. Now, T cells are also important, but that's a little more difficult to measure, but we're seeing more and more that this component, for some diseases, T cells are important to stimulate as well. But we want to stimulate the immune response, antibodies and T cells, to prevent disease. That stimulation produces a response by the adaptive immunity, but it does so without causing infection or causing disease. So that's the idea of vaccines, to stimulate the immune system but not cause disease. We will talk about traditional vaccines, and these include attenuated or inactivated, and that should be live attenuated versus inactivated vaccines. And then there are many approaches that we will talk about in terms of modern vaccines. And don't forget to appreciate that cute little baby on this slide, which I forgot to point out. Okay, here is immunology in a slide. We'll be covering this in the future. Today I'm focusing on vaccines, but you have to know a little bit about the immune response. Mainly, the immune response involves B cells, which produce antibodies. Those antibodies can bind to foreign invaders, such as viruses. They bind to specific regions called antigens, and they can either tag them for destruction. They can bind this virus to prevent it from entering another cell, and so they can prevent infection. T cells are also a part of the immune response. Cytotoxic T cells will actually destroy virally infected cells. They recognize antigens presented to them, they recognize that cells are infected, and they destroy them. The important thing with vaccines is that the immune response and some of these B and T cells become long-lived memory cells. So they remain in the body, and they are specifically designed to attack a specific type of pathogen. So these remain in the body so that next time this individual encounters the exact same virus, these cells will be stimulated immediately, and they will begin to attack and protect the individual. This response, the first time a virus or pathogen is encountered, this response can take up to weeks to develop. But the second time that same pathogen is encountered, the response is immediate because of these memory cells. So that is the basis for vaccines, to stimulate a response that will then produce these memory cells so that next time an individual encounters the actual pathogen, the live infectious agent, they will respond immediately and clear that agent from the body before any disease develops. All right, pretty good. One slide, immunology. So moving to traditional vaccines, there are two types of traditional vaccines. The first one is live attenuated viruses, actually I cover these in reverse order, I now realize. And so live attenuated virus or inactivated virus. There are disadvantages to these types of vaccines that we'll see as we're covering them. The viruses in both these cases need to be cultivated, so that can be time consuming, involve a lot of resources, and be labor intensive. Production, if you need large amounts of viruses to vaccinate lots of people. Yields can be, if yields are low, again, that takes lots of material and time and labor. Maintenance of those stocks of vaccine is also required. Usually they need to be kept at cold temperatures, which can be a problem in some countries, and there can be side effects using these traditional approaches. This is a list, and this is from the book. I don't have an updated list, if there is one. So it's from 2016 from the textbook, Vaccines in Use, and just run through them. You can see that all these listed here are either inactivated or live attenuated virus vaccines. Looking at inactivated viral vaccines, their first traditional approach. The virus is isolated, it is grown, and then it is inactivated by, in this case, formalin. So this is formalin inactivated. Inactivation, since viruses are not alive, we don't consider them being able to be killed, but we say that they are inactivated. So when we inactivate these viruses, they are no longer able to infect cells. They still have the same viral structure. They still have those same antigens on the outside of the virus that can be recognized by antibodies and T cells, but they have lost the ability to infect host cells. Okay, that's what inactivation refers to. My examples of both of these are poliovirus vaccines. These are some of the first vaccines that were developed using these methods. The first poliovirus vaccine that was licensed was the Salk vaccine. And here we see Jonas Salk immunizing a child. This is a formalin inactivated virus vaccine. This is still used when it is used. It's still used in the United States. And we're going to take a look at another type of poliovirus vaccine. It is most costly between the two types of poliovirus vaccines, but it's still used in the U.S. Because it's still used because it cannot cause disease. So we'll compare that with the live attenuated form coming up. So live attenuated viral vaccines, these are viruses that are isolated. And here we see these are human cells shown in orange. This is we'll call it the polio virus. That'll be our example. And we can see that these are able to infect the human cells. These viruses would be isolated from a patient and grown in human cells, and then they would be moved and cultured in monkey cells. Over time, in those monkey cells, they will develop mutations that will allow them to adapt to monkey cells. So over time and over many passages from one monkey cell line to another, more and more mutations are acquired that allow these viruses now to grow very well in monkey cells. They have, however, looking at this last panel, they have lost the ability to grow in human cells through those passages in monkey cells. So they are live, they're still able to infect host cells, but they no longer can infect human host cells. So now they can be used as a vaccine. So the second type of poliovirus vaccine was developed by Albert Sabin. It is considered the Sabin vaccine. This is an oral vaccine, and it is live attenuated. It's trivalent and has three poliovirus types in it. So this image here shows each type of poliovirus and how it has been inactivated. You can see many passages through live monkeys, 24 passages through live monkeys, 43 passages through monkey kidney cells, etc., etc., purification, and now we have vaccine strains of this virus that are no longer able to infect human cells, but they can be recognized by the immune system. So each of the types of poliovirus has been inactivated in slightly different ways, and all of those are included in this Sabin vaccine. This is not used in the United States, but it is used nearly everywhere else in the world, and that is because it is least expensive, and it also easy to administer because it is oral. It doesn't require syringes. It doesn't require medical personnel to give the vaccinations. But the downside is that these viruses can and do occasionally revert to being able to infect human cells, and they can cause disease in rare cases. So those are our traditional vaccines. This is the same chart I showed earlier. Some of the disadvantages. Not all viruses can be cultivated in cell cultures. Viruses can be inadequately inactivated, or they can be inadequately attenuated, and then they can go on and still cause disease in the individuals who are being vaccinated. So that's not a good thing. Those attenuated live viruses can revert and infect cells. There can be low yields of vaccine strains, because think about if you have to grow enough virus to include in every vaccine dose, that's a lot of growing viruses. So if you have vaccine strains that don't yield many viruses per infection, then that's going to be a difficult process. There are shelf life limitations. Again, most of these need to be kept at cold temperatures. And frankly, not all diseases or viral diseases can be prevented. And the examples, HIV, hepatitis C, these can't be prevented using these methods. So now you can see what the traditional vaccines look like. Now we'll start looking at new developments, new approaches to designing vaccines. These are possible because of recombinant DNA technology. They are usually safer because rather than using whole viruses, which is what we see in the traditional vaccines, we're using parts of the virus. So there's no possibility of reversion for most of these. And so I've listed here some of the ones that we will be going over. I think I took resortant viruses out. And we will go through these one by one. And then after I have a slide for each of these, and then I'm going to repeat the information, but focusing on coronavirus vaccines that are currently being developed. Okay. So recombinant subunit vaccines, and these can also be referred to as peptide vaccines. These are using a peptide, a protein from the virus in order to immunize. They're not using whole viruses. Okay, these peptides can be made using recombinant DNA techniques. So that's why they're called recombinant. That's why they're subunit, because it's a part of the virus, not the whole thing. I think that this term is more widely used now. Recombinant subunit may be outdated. The book uses that term, but I'm not 100% sure. But I think the peptide vaccines is a more widely used term for these types of vaccines. So how these are made is that we have a gene here that has been isolated for an HBV. This is hepatitis B virus surface protein. So this when it is expressed will form a HBV antigen. This is then cloned into a vector. Our example shows a yeast vector. So we've got our gene cloned into a yeast vector. These can be transformed into yeast cells, the host, and those yeast cells then can be cultured to obtain many yeast, a large population of yeast, and each of those yeasts are going to be producing our HBV antigen. So we're amplifying that antigen in yeast cells. Then after that amplification process, the antigen, the peptide, can be isolated and purified and used to vaccinate. Recombinant live vector vaccines. For these, genes that encode that antigen or that surface protein that is going to stimulate the immune response, that is inserted into a virus that is considered a safe virus. In this case, this is a vaccinia virus that will not cause disease in humans. Other examples of virus vectors are adenoviruses are being developed for live vector vaccines as well. So then this safe virus containing the foreign gene, the gene for our specific viral disease, can infect host cells, and as a result, we get recombinant vaccinia virus. So we get viruses that now contain that gene for contain that recombinant gene that will express proteins that will stimulate an immune response against the virus of interest. An example is a rabies vaccine. There's a rabies vaccine that has been developed this way. I believe it's used for veterinary use. Naked DNA or plasmid DNA vaccines. These consist of plasmids that have been manipulated. So this is not a viral vector, this is a plasmid, and it has our antigenic gene of interest inserted into it. This can be used as a vaccine. It can be injected into an individual. That DNA then enters cells, and those cells will recognize the plasmid and begin to express the antigen. So the host cells themselves express this antigen and present it to the immune system. Messenger RNA vaccines. Also a new approach. For messenger RNA vaccines, messenger RNA itself for a viral antigen of interest or a viral protein of interest, that messenger RNA is delivered into host cells. The host cells then express that protein, present it to the immune system, initiating an immune response. Now, messenger RNA, there are two types of constructs. There are non-replicating messenger RNAs and self-amplifying messenger RNAs, so that the self-amplifying messenger RNAs will produce a higher yield of antigen. You can't really just inject messenger RNA into individuals and expect it to get into cells. It will be degraded before it reaches the cells. So there is methods of delivering the messenger RNA also require development and are being developed. I think the main method is to encapsulate those in lipid particles, and those are considered nanoparticles. And so here we see messenger RNA has been encapsulated by a lipid membrane, and then that will enter the cells. There are issues with immunizing the compromised host. And so because of that, I'm not going to go into detail, but because of that, this is another, I should have put this with the disadvantages of traditional vaccines. Use of live vaccines is not recommended. Even, I believe the use of live vector vaccines is not recommended. Other vaccine issues. Vaccine additives. Are there things we can add to vaccines to stimulate an even greater immune response? Adjuvants sometimes serve this purpose as well, so developing different types of safe adjuvants. Minimizing side effects. With any therapeutic, there are side effects, so we want to make these as rare as possible. And then issues with vaccine delivery. So these are other regions of vaccinology that are being developed. Can we find new ways to deliver? Nasal sprays, skin patches, time-release pills. That needle and syringe system is not adequate to vaccinate the world. All right, when vaccines are manufactured, it is a long process. Traditionally, it is about a 10-year process, costing, I think, millions to $1 billion. Okay, so it's a long process. Lots of trials, clinical trials need to be performed before there is any FDA license approved. So let's go through this. And this is more than you will need to know for your paper itself, but for your information, it's good to know. So stage 1, this requires studies in animals to show effectiveness. And then FDA needs to approve moving into clinical trials, so starting to test these vaccines in humans. This is the second stage, right? So phase 1 is to use healthy volunteers, and this uses small number of subjects. And this is looking for dose ranges and safety. Oh, down here, yes, 2 to 4 years. Phase 2, then, we're starting to amplify the number of subjects, but it's still pretty small, 100 to 200 human subjects. Determining the dose, determining that it does cause an immune response, and also using placebo group in stage 2. Sorry, phase 2. Phase 3, we begin to see full-scale manufacturing of this. And now our subject numbers are moving into the thousands, and including subjects that have pre-existing conditions to determine whether this will be safe and effective on those individuals as well. Once a vaccine has passed through phase 3, then FDA license can be given if the results are adequate, and that vaccine moves into phase 4, where the vaccine is available to physicians and patients, and we move into much larger scale safety and clinical trials. Also, and always, officials never stop monitoring for adverse effects in vaccines. I'll just include this as well. There aren't that many companies that produce vaccines because daily pharmaceuticals are more profitable. So this idea that government tries to force people to get vaccines in order to benefit biochemical or pharmaceutical companies is not really true. If you look at how much it costs to develop a vaccine, and knowing that if you can develop something that people have to take every single day, that's much more profitable than a once or twice in a lifetime vaccine. The majority of individuals that get vaccines and need vaccines are in impoverished countries. So because of that, UNICEF pays for many of these vaccines in order to provide them for children in these countries, as well as the Global Alliance for Vaccines and Immunization. Okay, so economics of vaccines. Moving forward, I'm going to repeat some of the information on types of vaccines because we're going to look specifically at some SARS-CoV-2 vaccines and the approaches that are being used to develop a vaccine currently, today, going on right now. If you already have a great idea and you can stop now, it's fine. I'm just going to go through those types of vaccines again. I think it's interesting. We look at the vaccines being developed, and this is from this article right here. You can go take a look at that. That was published at the end of April, so there may be some new information, but most of it is still current. If we look at the types of vaccines being developed, those traditional types, inactivated or weakened virus, we see that there are less than 10 being developed, so that is the smallest categories, are traditional types of vaccines. Viral vector vaccines, we see a lot of groups working on viral vector vaccines. Nucleic acid vaccines, that includes the DNA vaccines and RNA, those messenger RNA vaccines. And protein-based, we have our largest number of companies working on protein-based or peptide vaccines. So we've got protein subunit, that would be peptide vaccines, virus-like particles, which I did not cover. I didn't cover those. All right, lots of vaccines in development. Okay, the traditional approach we've gone over. The weakened virus, so these are live attenuated virus. Let's see, there is the companies working on this, Codagenics in New York. Okay, it doesn't say how many. Some of them it tells how many companies are working on that, but we know how these are made, right? Inactivated viruses. There are some companies working on making inactivated viruses as well. Inactivating those viruses by using chemicals such as formaldehyde. Heat can also be used to inactivate viruses. And again, these are no longer able to infect cells, any cells. They are inactivated. Viral vector vaccines, or recombinant vector vaccines, which is what I called them earlier. There can be replicating viral vectors used or non-replicating viral vectors. So a safe virus is used to vaccinate one that will not cause disease, and in that viral genome is inserted, for our coronavirus vaccine, a coronavirus spike gene, so that when these viruses replicate, they will present that spike protein to the immune system. DNA or RNA vaccines. Here are our DNA vaccines. We see the coronavirus spike gene inserted into a DNA plasmid, and then that is injected, taken up by cells, and those cells then are going to express that coronavirus spike protein themselves. That protein will be detected by immune molecules, and an immune response will be formed against the spike protein. We see similar thing happening with RNA, except that again, that RNA is encapsulated in a lipid molecule that will then enter cells. So you're sort of skipping a step, actually. If you look at the picture, we're skipping the DNA step. We're going with just messenger RNA. Protein-based vaccines, protein subunits, peptide, recombinant subunit vaccines. 28 teams are working on these vaccines. And so that I showed you how these are made in a recombinant manner. We saw the example of using a yeast plasmid, amplifying the production of this protein in yeast, and then isolating and purifying that. Then those spike proteins are used to make up the vaccine. Those proteins themselves will be injected into individuals and recognized by immune cells and result in an immune response. Virus-like particles I did not go over, but because of the ability of viral structural proteins to self-assemble, you can make those structural or express those structural proteins, and for some viruses they will self-assemble into an empty virus capsid. And there's no genetic material here, so it cannot cause any disease. It cannot infect cells, but it can be taken up by the immune molecules, recognized as foreign, and now we have an immune response against the surface structure of coronavirus. The notes up here indicate that while it can trigger a strong immune response, these can be difficult to manufacture. There are a couple vaccines that are prepared this way, though, and hepatitis B is one of them. So this is just going through the approval process again. I won't go through that first part again, but I do want to point out Warp Speed, which is the U.S. government's program, and they will name 5 or more vaccine projects, and they've already been giving money to some of these projects to fund them and to get some of the manufacturing process, which takes a lot of time, get that going along with the phase 2 and phase 3 trials. So having multiple steps being performed at the same time to reduce the amount of time it takes to get to a vaccine that is approved. All right, let's move forward. I have included this NewYorkTimes.com link at the bottom. This is a coronavirus vaccine tracker. So you can see the last update was just this week, and you can keep track of vaccines that are being developed against coronavirus. You can see a lot of them in phase 1, fewer in phase 2. There are actually 3 vaccines approved for use for early or limited use. One of these is in China, one might be in Russia, I can't remember. But there are still no approved vaccines. Of course, it's been less than a year, and it does take time, and we want to make sure that those vaccines are safe and effective. So that's all that I have for now. I leave you with this cute baby who has just had his one set of vaccinations. And I hope this is helpful with you, giving you some ideas of vaccines that are being developed and that you can move forward in your writing assignment. If you have any questions, feel free to ask or email. Take care.